let's look at a genetic code table, this one, and you will notice that there is a stop sign here, UAA, UAG, UGA, and we had UGG as our wild type gene. So UGG codes for the amino acid tryptophan. If that G mutates and it becomes an A, okay, so the G has now mutated to a UGA will now code for a stop sign. So now what is going to happen to that protein, to that amino acid? There is no amino acid, so it will become a missense or a nonsense code. So a nonsense mutation changes the normal codon into a stop codon. Okay, now let's look at this example. These are not nucleotides, these are just words, but what if similar things would happen? Okay, so if these letters are arranged the right way, then we can of course read them. It's English language. The big bad dog ate the fat red cat. If there is a missense mutation, can you see where the mutation is? So it's right here. The A has mutated to I, okay? And it changes the whole meaning of that sentence. And it'll cause a different amino acid to be put in that protein. In silent mutation. So in silent mutation, the base changes, but it does not change the amino acid. Now, how is that possible? Let's look at this. So if you have, for example, AAU as your wild type gene, and the U mutates into a C, can you see what will happen? U mutates to a C. Will there be a change in the amino acid? No. They are both coding for the same asparagine as the amino acid. It could happen, let's say, up here. This is the wild type gene. It codes, or it reads UCU. Now, what would happen if the last U, this one, changed into, what could it change to? G. Can you see what will happen? If it changes to a G, it'll still code for serine. So it's called a silent mutation. So it can be weird like that. I'll give you one more. CGU. Again, U mutates to G. Okay, so watch this. U mutates to a G, will still code for arginine. Okay, now what if the wild type is CGG and that C mutates to a A? Watch, watch, watch. Mutates to a A. It becomes AGG, still codes for arginine. Okay, so those are called silent mutation. Alters the base, but will have no effect on the amino acid. Back mutations are two mutations, one after another. When a mutated gene reverses back to the original. So let's say UGG was the original, it went back to UGA, and then it mutated a second time and went back to UGG. That's back mutation. Frame shift mutation is when the reading frame is changed. Okay, so let's look at this guy again. The big bad dog ate the fat red cat. Frame shift mutation. Remember, in a frame, you're always reading 3 at a time. Okay, 3 letters at a time. if you have a deletion, one base gets deleted, okay? This guy got deleted. The A is out. You're still going to read it 3 at a time. So you're going to now read the big. The A is gone, so it'll be BDD, then OGA, then TET, then HEF. So see how every amino acid from then on, from this point on, will be different. This is going to read a different amino acid, different, and that's not even complete. So a deletion will shift the frame for every amino acid from that point down. What if you're not deleting a base, you are inserting a base? Okay, so these are both point mutations, one base changes. If you're inserting a base, a wrong base gets inserted by mistake. So a B gets inserted here between the BA and the D of the word bad. From this point on, you are going to read every amino acid codon different. So this will become BAB, DDO, GAT, ETH, etc., etc. So every single every single amino acid will be different from here on. Okay, so that is frame shift. Reading frame of the mRNA is completely altered, and everything is changed. So you will definitely have a new protein that most of the time is non-functional. Okay, now when you see that moose change into an albino moose, what exactly happened? The enzyme that was supposed to make the pigment for its skin or coat or hair color did not get made because it was non-functional. And any or all of these could be responsible for that. Do mutations get repaired? Some do and some don't. And this will bring us to our RNA viruses and DNA viruses again. So because mutations are fatal, some mutations can kill you, right? For example, if you have a mutation in your mitochondria, you're dead. You're not going to survive. If bacteria cannot copy its DNA, the gene for copying it is mutated, DNA polymerase is mutated, it's dead. So some mutations are fatal. To stop that kind of mistake happening through mutation, cells have repair mechanisms in place. If a mutation happens, DNA polymerase can do another thing. Beside copying DNA, it can do a second function. So yes, DNA polymerase copies DNA to DNA during DNA replication, but it has a second function. It can also proofread. So it can go back, it can check, and if it has put in a wrong nucleotide, for example, it's supposed to put AAA across from a T, right? It's a complementary base. So it's reading one strand. Let's say it's reading this strand, and it's making this strand. It's supposed to put AA. But let's say by mistake, it has not put an A in there. It has put a T in there. DNA polymerase can come around. It can read this and see that there is an error in it. It will take that base out and it will put in a new and the correct base again in there. Okay, that's proofreading. So it can locate the mismatched pair. Light is also a repair mechanism. So if somebody sits in the UV light and gets a lot of DNA mutated, so some people sit in the tanning beds and get UV light damage to their skin, cells are mutated, DNA is mutated. Regular light, visible light, can come in and repair that damage sometimes. So most of the time people don't get cancer because there are repair mechanisms inbuilt in your cells. There's another method called accession repair, and here enzymes will go in and remove that entire segment of the DNA where the mutation is. And then DNA polymerase again comes in and it will add the correct nucleotides back in again. So some little provisions are in place for these things. Next big thing we will look at is called Ames test. So Ames was a biologist who discovered or figured out this test by which any chemical can be tested. And the question that they ask in this test, that's the big question, is this chemical mutagenic? Or can this make our DNAs behave weird and change? Oh, wrong spelling. It's terrible to write with a mouse, but here it is. Genic. Okay, so this experiment will answer that question. Every single day we come out with new chemicals for agriculture, industrial use, medicine, etc., etc. And the federal government runs Ames test on every single chemical, and no chemical is released into our society for human consumption or use without doing this test. The organism that this is tested on, the test organism, is Salmonella. So that's a bacterium, right? So you all know that this is a bacterium, and this is not a normal Salmonella. It itself is a very strange mutated Salmonella. So we do not use a normal Salmonella. We use a mutant Salmonella. What kind of mutation does it have? It has lost the ability to make histidine. So histidine is an amino acid, and every organism needs this amino acid because every organism has proteins in their body that will need this amino acid. And most organisms can make the histidine. Definitely normal Salmonella can make the histidine, but this Salmonella cannot. So this Salmonella is called His minus strain. This strain cannot make histidine, so we call it histidine minus. You start with a culture of this Salmonella strain, this mutant Salmonella strain that cannot make histidine, and you take agar plates. These have very special media in them. They're called minimal media, and the minimal media has no histidine in it. Okay, so it has everything else that the bacteria can use for growth and cell division and for making colonies, but it does not have histidine. And this medium on the other plate is the test plate. So this one is a control plate, this is a test plate. This test plate also has minimal media, but it has a test chemical added. So whatever test chemical we were going to be using is added to that. Okay, so let's say I wanted to test bleach, and I asked the question, is bleach mutagenic? Then I would add bleach on top of this. Okay, the Salmonella is then added on to top of the control plate and is also added on top of the test plate. Now, the test plate also has no histidine, okay? So there's no histidine anywhere, but histidine is very important for the bacterium to actually grow. So what's going to happen? Bacterium without histidine doesn't grow. So most of the plate is going to be empty. There'll be no colonies here. And once in a while, you'll get a few colonies. So you can see 3 green colonies here, 1 little colony. Once in a while, you'll get colonies because of the spontaneous back mutation. So by spontaneous mutation, some colonies are changing from His minus to becoming His plus. Okay? They are becoming normal wild type cells. And those few colonies will grow. What is happening on this plate? If the bleach is mutagenic, it's going to change and mutate the bacterium. And if it mutates the bacterium from His minus, it'll go back to His plus. And then all the colonies will start growing. Okay? And that tells us, so we look at this plate and we say, oh, colonies growing, which means they have all become His plus now, which means my test chemical, which was bleach, is mutagenic. It's causing mutation in the His minus. So that's how cool it is. But what if there was no colonies here? If there are no colonies here, then we would say that test chemical is not mutagenic. Once in a while, you will have same result on this plate and that plate, that in the sense that there'll be those 3 colonies here from back mutation by spontaneous, okay? So the degree of the mutagenicity cannot be calculated, but you can just say yes or no. Are they mutagenic or are they not? If mutations happen and they make non-functional proteins, proteins that do not work like they should normally, then that can be harmful. And if these important proteins are not working properly, it could be fatal to the cell and the organism. Okay? On the other hand, there are organisms that have mutations and they actually do better. The mutations are good for them. Maybe it helps them adapt better. Maybe it helps them survive better. Sometimes they help them reproduce better. And this actually causes change in the population, right? So if you have a white moose in a brown forest, it's going to stick out like a sore thumb and be visible to its predator. But if you have a moose whose coat color is camouflaged against its environment, it'll survive better, right? That kind of thing. If the polar bear who are drowning right now in the liquid cold waters of the Arctic Circle because our glaciers are melting, if they cannot survive, and then one bear is mutated, and that mutation causes that bear to have webbed feet, and then the polar bear can swim really well, especially the little cubs can swim really well, that will change them. That's going to change the population. They're going to survive, right? So any mutation that gives them an advantage will be saved and retained and kept and maintained in the population because those are the organisms that will actually survive.